Today we will introduce an iGEM team from 2019.
2018.
18, oh sorry.
Yeah, it's fine.
It's not 18, okay.
I would like to introduce FAU-GEM.
Hello and welcome to the fourth episode of our podcast FAU-GEM, where we introduce you to different iGEM teams that we found to be interesting. My name is Maria.
And I'm Feyza.
And we will be today's hosts. So maybe we will start with the team and the project. We will introduce the team from 2018. And Feyza, do you want to tell our audience about that?
We will talk about the iGEM team Hamburg and their project called Reagents of S.H.I.E.L.D, which is a clever reference to the Marvel team series Agent of S.H.I.E.L.D.
Yes, and during the competition, the team has been awarded some awards. Once, on the one hand, one for the entrepreneurship, because they reached out to companies to discuss the potential for commercialization of the device, and even filled a preliminary patent for it. And also they identified the economic potential of their project in general, which is why they got that award. In addition to that, they also got an award for the hardware of the device, and also one for the modeling, because they built a new model for protein synthesis, including DNA, RNA, and protein species. And also they got a gold medal. And to introduce the team consists of 16 students from the universities Hamburg and from the Technical University Hamburg. And so it's a German team. And their field of study is they have students from different fields of study. They have biology students, nanotechnology students, but also students from molecular life sciences, and also chemistry and engineering. So it's really interdisciplinary. The team itself is a overgraduate team. And do you want to should we start about a project? I think that's fine, right? The problem they worked with is malaria. What can you tell me about malaria?
Malaria is an infectious disease. I think it's caused by mosquitoes.
Yes, that's right. And it's a really deadly disease. It causes over 440,000 deaths per year. And it's caused by Plasmodium parasites, which are transmitted to people through mosquito bites. But not all mosquitoes are responsible for it. Only infected mosquitoes from the Anopheles genus can transmit the disease. And those specific mosquitoes are present on every continent, but especially in tropical areas where they have the best conditions to reproduce in a fast way. Generally, for the reproduction, a female lays something between 50 and 200 eggs directly on water every time they oviposit. So the problem with that is that they can oviposit several times during their lifetime, and their lifetime consists out of I think a month. So that's a lot of mosquitoes that come from only one. And the most malaria cases are in poor regions of Africa and Southeast Asia, because they have a lack of infrastructure there. And also, in addition to malaria cases, they have other humanitarian crises. So that also contributes to the severe situation. And we already talked about transmission for a short while. Do you know how the mosquitoes get attracted to their targets?
I think I heard about that. Mosquitoes are attracted to their targets through various chemical compounds, such as carbon dioxide or lactic acid.
Yes, exactly. And those compounds, CO2 occurs because people breathe, and lactic acid can be found on human skin, I think. And after biting their target, the parasite from the mosquito saliva passes into the bloodstream of its victim and reaches the liver, and then it mostly stays there and reproduces. And a problem with that is that it's mostly invisible to the immune system because the parasite itself spends most of its lifetime in the liver and in the red blood cells. And symptoms of malaria infection are flu-like symptoms like headaches, chills, and after the illness has progressed, also severe anemia and renal failure. And because it's such a severe illness, of course, some approaches have been done against malaria, mostly in the use of bed nets and vaccines, but also with development of anti-malarial drugs and insecticides. And I mean, both of us come from a chemical background. Do you have a specific insecticide in mind that was used for such a cause?
I read about it. It was DDT.
Yeah, that's right. DDT was used up until the second half of the last century against malaria, or against the mosquitoes that transmit malaria. But a problem with that is that it's a persistent chemical, so it does not really get degraded in nature. Instead, it accumulates, and it accumulates also in the food chain, which is a problem for humans when they eat animals that have a large accumulation of DDT. And DDT is lipophilic, so that means that when it accumulates in the fat tissue, and there it leads in the human body itself. It leads to it's an endocrine disruptor and leads to a higher rate of abortion in pregnant women, and also is suspected to be carcinogenic. So it's really not a nice compound to have in your body, which is why it's not used anymore, at least in the US and Europe, I think. And okay. And since the mosquitoes spread so fast, other methods that work against or that combat the reproduction of those mosquitoes are needed. And one of those devices is the S.H.I.E.L.D. device from our iGEM team, Hamburg. It's a device that's used to fight malaria transmission, and it counts as environmental synthetic biology project. So now we can introduce the project. It's called S.H.I.E.L.D, and it's an abbreviation that stands for Sustainable Human Imitating Elimination and Lure Device. And what it does is that it lures and kills the vector of malaria transmission, so it would be the mosquitoes in this case. And do you know who would specifically benefit from such a device like S.H.I.E.L.D?
Yes, I know that it's especially beneficial to high-risk groups such as pregnant women and children under 5 years old.
Yes, because those groups cannot take most malaria medication because it has many side effects. And so the project itself is a great way for those people to be protected against malaria to a certain extent, I think. And on the wiki, the team described their project as a hardware that encloses E. coli bacteria while also balancing the containment and also as having a selective connection to the environment. The device itself is self-sustainable and self-preserving, which means that it works for extended periods of time without needing maintenance, which is especially good for regions with poor infrastructure, like the regions where malaria is most common. And in the device, synthetic biology is complemented by a spatially separated co-culture and a condition-responsive hydrogel. And so the device itself consists of two parts, one upper part and a lower part, like this, roughly. I mean, go visit the wiki if you want to know more about it, because they have a great animation about the device itself. I can really recommend it. And in the lower part, we have the genetically modified E. coli bacteria, and what they do is they are modified in such a way that they produce heat, of course, but also lactic acid, myristic acid, and 3-methylbutanol, and all of those compounds are said to attract mosquitoes. And those are volatile compounds, and they are found on the human skin. And they lure the mosquitoes to the device. Also, the bacteria express a toxin. It's called black scorpion alpha insect toxin BJAIT, and this toxin is deadly for insects. And I already said that it's a cell culture that's enclosed in this device. Can you imagine what would be a problem with working with bacterial cell cultures in such a setup?
I think one major concern would be the depletion of nutrients in the cell culture medium.
Yes, because you're right, definitely, because normally when working with cell or with bacterial cell culture, the medium has to be changed regularly, and of course this would not be possible in such a setup. But I think they came up with a good way, or even a great way, of circumventing this problem. So what the team did, they also put up cyanobacteria culture in the upper part of the device, and it's transparent, this part of the device. And then with the help of sunlight and water, the cyanobacteria can produce glucose through photosynthesis, and the E. coli bacteria can use this glucose to sustain themselves. And so this means that roughly that the device can be sustained only by sunlight and water. And also, another problem that might occur is bacterial overgrowth, but this is prevented by the addition of a growth inhibition module. And this module also ensures the biosafety of the device, because it prevents uncontrolled proliferation of the bacterial cells, even in case of damage of the shield. So it cannot proliferate in the environment. And so how does it work? In general, we also have an hydrogel that covers the lower part of the shield device with the E. coli containing compartment. And this hydrogel provides a surface for the mosquitoes to land on and to sting into. And also a secretion module and an automembrane protease are present, and those two lead to the excretion of the attractants, so of the molecules I mentioned before, and the toxin to the hydrogel. And then when the mosquitoes sting into the hydrogel, they ingest the toxins and they die. Besides that, the hydrogel has self-healing properties, so even after the mosquitoes sting into it, it's ensured that no bacteria can escape the device. We then also have a nanofilter that's used as an extended barrier. So it extended the barrier that's already present or that was already created by the hydrogel between the genetically modified organisms and the environment. So that's it for the project part. Now we will continue with the collaborations. Feyza is going to tell our audience about that now, right?
Yeah, of course. The iGEM team collaborate through meetups, surveys, social media challenges, and partnerships with other teams like Eindhoven and Düsseldorf.
Yeah, and do you know what they did with Düsseldorf?
Yes, they provided the Hamburg team with the necessary theoretical knowledge to work with cyanobacteria.
Yes. So it's a really important part of the project. Also, another collaboration. Again, you can find all of them on the wiki. Another important collaboration was with the iGEM team Eindhoven. So the iGEM team Eindhoven developed a dextrane-based gel which immobilizes the bacteria by the expression of matrix-adhering membrane proteins. And this gel was used as matrix for the E. coli in S.H.I.E.L.D. So maybe to explain some things, immobilization of bacteria means that the bacteria confined in a limited physical space, and it just means it just explains the confinement of a viable microbial cell culture. And such immobilized cell systems can be used in or have been used in biotechnology for bioremediation and similar purposes. So it's really interesting, I think, because I did not know about it beforehand. Also, maybe you're asking yourself, what's dextrane? Dextrane is a polysaccharide, and it can have varying lengths between 3 and 2000 kilodaltons, and it's derived from glucose condensation and occurs, for example, as a microbial product in wine. Also, it plays an important role as medicine, and it's even part of WHO's model list of essential medicines. And you probably know it, but this list contains medicines that are considered as most important medications in a health system. And I think dextrane acts as an antithrombotic agent, or also is used to reduce the blood viscosity. In addition to that, it is also of use in the laboratory, where it is used to coat nanoparticles to prevent their oxidation, or also in size exclusion chromatography, where it's present in the matrices, and also it's used as an immobilization agent in biosensors. So it's a chemical with a broad application area, and it's also used here in this gel. And then, with the help of this gel, the team Hamburg obtained a 3D gel-embedded culture that ensures an even cell distribution of the bacterial cells throughout the culture, and also has properties that further inhibit bacterial growth. And then, as a part of the collaboration, they also looked at conditions that would lead to the dissolving of the dextrane gel. Can you imagine why they did that?
It is crucial to consider biosafety.
Yes, because we work with genetically modified organisms. And I mean, they already did that before, when they made sure that the gel has self-healing properties and stuff like that. But also, they wanted to see under what conditions or under what potential conditions the gel wouldn't do its job anymore. And for that, they looked at acidic conditions and at basic conditions. For the acidic conditions, they put the gel into hydrochloric acid, which had a pH of 3.0, and also they put it into lactic acid, which has a pH of 3.6, and then for the basic conditions, they put it in sodium hydroxide and in a commercially available compound that dissolves gels. And what they obtained from those experiments, again, they are described in greater detail on their wiki, is that the gel was stable even under conditions that exceeded the tolerance of most common bacteria. So this means that even if the gel were to be dissolved, the bacteria would not survive in the environment where this would be possible. So there's no real hazard from a potential spread of bacteria. And so that's really interesting, I think. And with that, we are already at the end of our podcast. You can find the sources in the description, I hope. And if you want to, you can follow us on Instagram. Our handle is
iGEM.erlangen.
Yes, thank you. Thank you for hosting the podcast with me, Feyza. I had fun. And
thank you, Maria, and thank you all for joining us today.
Yeah. See you or hear you next time.
Bye.
Bye.